Hello, welcome back to Bioclass Bites. In this video, we are going to talk about the central dogma of molecular biology, specifically DNA replication. Don't forget to subscribe to my channel and like and share this video. This video is part of Grade 8 Biology unit on the central dogma of molecular biology. These are the lesson titles for this unit. So we start off with DNA replication and then how DNA is transcribed and translated to make proteins and what could possibly go wrong once DNA mutates. In 1953, the following scientists, James Dewey Watson and Francis Harry Compton Crick, co-authored an academic paper wherein they proposed this double helix structure of the DNA molecule. What we know or what we visualize whenever we think about DNA. So during that time, it was actually a revolutionary discovery because before then, these are the things that scientists know about the DNA. They know that DNA is a weak acid. They know that a certain particle is involved in inheriting specific traits in organisms. Scientists know that genes are found in the chromosomes and that chromosomes are found inside the nucleus. They know that genes eventually code for proteins and that DNA is ultimately the genetic material of organisms. But despite all of those discoveries, they really have no idea how the DNA looks like. So when Watson and Crick, together with Wilkins, proposed this double helix model of the DNA, they were awarded the 1962 Nobel Prize in physiology or medicine for their discoveries about the molecular structure of DNA and its significance for information transfer in living material. Okay, so that makes their discovery, their work very important and very significant. So this is where we can find the DNA. So all living things are made up of cells. Okay, so if you're just one cell, you're a unicellular organism. So if you are made up of several cells, you're a several cells to trillions of cells, you are a multicellular organism. So in every cell, there's an organelle that contains the genetic material. We call that the nucleus. Inside the nucleus are your condensed genetic material. Okay, so if your DNA is condensed or tightly coiled, they form your chromosome. Okay, we've talked about this in the previous unit on cell division. Now, your chromosomes are actually tightly coiled DNA strand, okay, tightly coiled around proteins called histone. Okay, so these are your histone proteins, these small globular structures here. And if you unwind that DNA strand, okay, this is how it looks like. Okay, so it's a double helix. Double means because it has two strands. Then helix because it's wrapped around like a circular ladder in that structure. Now, if you zoom in closer, you will see that your DNA is actually made up of this backbone of sugar and phosphate with the base pairs that makes up the ring of the ladder or makes up this part of that ladder. So again, your DNA strand coils around histone proteins, and then they are actually condensed and tightly packed in chromosomes, which makes them easier to be divided during cell division. And then your chromosomes are found inside the nucleus of the cell, if you are a unicellular organism, or in all the cells of your body, if you are a multicellular organism. Okay? So that's the DNA structure found inside the nucleus. Now, the following videos are not just recommended but required for you to watch. So this is from Stated Clearly, what is DNA and how does it work. So it will explain to you the double helix model of the DNA. So it will give you a lot of information about it. So I'll provide the link in the description below. This one is another video, the structure of DNA. Again, I'll provide the link in the description below. So these two are required videos for you to watch. I also recommend that you visit this lesson from Khan Academy, overview of gene expression. So I'll provide the link in the description below. And then this video, which will summarize everything about the central dogma of molecular biology from DNA to protein in 3D. Again, another required video for you to watch. I'll provide the link in the description below. Now let's compare the following molecules that are very much involved in DNA to protein processes. So you have your DNA and then your RNA. So DNA actually stands for deoxyribonucleic acid. D from de, N from nucleic, A for acid. Okay? So RNA on the other hand stands for ribonucleic acid. R N A. Okay? So these are their differences. So the first one is that DNA is double-stranded. It has these two strands of sugar and phosphate backbones with the base pairs inside. So one strand, two strands. RNA, on the other hand, is just single-stranded. So you just have a backbone of sugar phosphate with a single nucleobase attached to it. Okay, so DNA double strand, RNA single strand. Next, the ribose structure, okay, the sugar structure of DNA. So they are almost the same, except that at this part, okay, it is missing one oxygen. So as you can see here, they're almost the same, but here, RNA has an oxygen attached to this hydrogen. But here in DNA, it is missing one oxygen. That's why it's called deoxy. De, non-oxy, oxygen. None or no oxygen or missing one oxygen. So that's another difference. Okay? And then in terms of nucleotide bases or nucleobases, they both have cytosine, guanine, and adenine. So those are the base pairs here. Okay, the bases. Okay, nitrogenous bases that you can find in the rings of the ladder. So they both have cytosine, guanine, and adenine, but DNA has thymine and RNA has uracil. So there's no uracil in DNA, there's no thymine in RNA. So these are more differences of DNA and RNA. So again, DNA is double-stranded, RNA is single. They both have, yes, and then these are structured into chromosomes. Then the sugar for DNA is deoxyribose. It is missing one oxygen. For RNA, it has a ribose sugar. It uses thymine. It uses uracil. The function is for genetic information for DNA. And then this is mostly for protein structure, or protein synthesis. Can self-replicate, and then DNA can self-replicate. RNA have multiple structures, mRNA, rRNA, and tRNA. DNA can last for a very long time and are actually inherited. RNA does not usually last for a long time. And it's read from 3' to 5', and it is formed 5' to 3'. This will be thoroughly discussed in the videos I mentioned a while ago. Now they still have similarities. They both have, I've mentioned this, they both have adenine, cytosine, and guanine as their nitrogenous bases, and they both have sugar and phosphate backbone. Sugar and phosphate backbone, okay? The sides of the ladder, if you want. So again, no, DNA is a molecule composed of two chains that coil around each other that carries the genetic instruction for the development, functioning, and growth of the organism, while RNA is mostly for coding and decoding and regulation of expression of genes, and it is important specifically for DNA and protein synthesis. So what does the central dogma of molecular biology tells us? It says that, very, very easy, the DNA that's found inside the nucleus of every cell are copied and translated by RNA to make proteins. That's it. The central dogma of molecular biology tells us that the flow of genetic information is from DNA inside the nucleus, transcribed and translated by RNA to make proteins in the ribosomes in the cytoplasm. Now, you might be thinking, oh, that's not a big deal. Okay, we know DNA, then RNA to make proteins. But what if I tell you that these processes are done by all the living things in the planet, starting from a microscopic bacterial cell into something as big as a blue whale or even dinosaurs, even humans. All living things share the same processes. That's why it's a central dogma. Dogma means principle or tenet or belief. Central dogma of molecules because DNA and RNA are molecules. Central to biology, to all living things. This is something that unifies all living things because this is the process by which genetic material is copied and how it makes up proteins that eventually is the basic structure of all living things. Okay? So in bacterial cells, the DNA replication, transcription, and translation all happens within the cell because they do not have a nucleus. They are prokaryotes. They do not have a modern nucleus. But for eukaryotic cells, the DNA replication and transcription happens within the DNA, while the RNA translation and protein synthesis happen in the cytoplasm. So again, what makes this a big deal is that this process is similar for all living things, from something as small as a bacterial cell to something as big as your blue whales and elephants and giraffes, from something as simple as a prokaryote to something as complicated as the human brain. All cells, all living things, they have these processes. Where does it take place? So DNA replication in eukaryotes happen inside the nucleus. As you can see here, this is the nuclear membrane. Okay, and then the DNA is transcribed or copied by the RNA because the protein synthesis takes place in the cytoplasm. The information that codes for protein synthesis is in the DNA. So the DNA cannot leave the nucleus, okay? It's protected by the nucleus. Therefore, something has to copy the information. That's where the RNA comes in. So the RNA copies the information from the DNA, then it goes out through the nuclear pore. It goes out into the cytoplasm, into the ribosomes, carrying with it the genetic information needed to make the protein. Okay? So DNA replication inside the cell, transcribed by the RNA, then brought out of the nucleus to be translated to make into protein. So this is the central dogma of molecular biology. So again, this process of central dogma involves DNA replication, okay, RNA transcription, and then RNA translation, and then eventually for protein synthesis. Now, what makes it important? It forms the basic storage, transmission, and expression of hereditary information. You might take it for granted, but do not forget that we are mostly made up of proteins. Our muscles are made up of proteins. We have keratin in our hair, hemoglobin in our blood. Some of our hormones, some of our enzymes are made up of proteins. Some of our most of our structures are made up of proteins. So basically, we are just giant proteins. And all of those information that codes for making proteins are found in our DNA. So That's quite a big deal. Okay? This discovery is quite a big deal. Now, I recommend that you visit this website. It shows us the timeline of DNA discovery. So you can actually click the timeline and see what are the major discoveries during those periods. Okay, I'll provide the link in the description below. So for this unit, we will go through the different processes of central dogma one by one, okay? But for this video, we will start first with DNA replication. Now, how DNA replication happens in its smallest details, I recommend that you watch this video, DNA replication fork, ADH animation. I'll provide the link in the description below. So if you still remember our lesson from the cell cycle, specifically during interphase, DNA replication happens in the S phase, okay, of the cell cycle of eukaryotes, okay? So the S here means synthesis phase, okay? That's the time that DNA is replicated. And to read more about this, I recommend that you visit this lesson from Khan Academy. I'll provide the link in the description below. So there's two important concepts we have to clarify whenever we talk about DNA replication. First, that DNA replication is semi-conservative and it is semi-discontinuous. Okay? Semi-conservative because um, the daughter DNA is composed, or the resulting daughter DNA is composed of one of the two parent molecules, or one of the two original strands, and a newly synthesized, a newly created complementary strand of that molecule. So again, it's not conservative, wherein um, the entire parent DNA is copied. It's semi-conservative because only half of the new daughter molecule is is directly coming from the parent DNA. The other half is a synthesized or a created copy of that strand of molecule. So that's the first one. So we will return to semi-discontinuous later. So again, DNA replication is semi-conservative, meaning that each strand in the new DNA, each strand in the DNA acts as a template for the synthesis of a new complementary strand. Okay, so yeah, so like what I've mentioned, no, it takes us from one starting molecule to create two daughter DNA molecules with each newly formed double helix strand containing one new and one old strand. So one new is the newly created copy, then the old strand is the original parent strand. So that's why it's called semi-conservative. Half, semi, half, okay? Half is conserved from the original parent molecule. Now we found out about this. We know about DNA replication being semi-conservative thanks to Matthew Meselson and Franklin Stahl, because they conducted an experiment which heavily supported the Watson-Crick hypothesis or Watson-Crick model of the DNA. In their experiment, they proved that the DNA replication is actually a semi-conservative process. It's what's actually correct. And because of that, it was considered the most beautiful experiment in the field of biology because they were able to figure out how DNA molecules preserves its original identity while still capable of making new copies of itself. Okay, so I require that you watch this animation about the Mendelssohn and Stahl experiment of DNA replication. So I'll provide the link in the description below. So basically, this is what they did. Okay, so they used the DNA molecule of E. coli. Okay, and in the the first generation, they used a medium with an nitrogen 15 isotope, so that this was absorbed by the DNA of the E. coli, and then they also exposed it into a lighter isotope, nitrogen 14 isotope. Okay, and then after that, they allowed the E. coli to undergo cell division, and they were able to track down how much of the DNA molecule containing the N15 isotope is transferred to the next generation, and how much of the N14 um, how much of the DNA having the N14 isotope is carried to the next generation. And this is what they found out. So, and then they exposed the cell. They extracted the DNA molecules of the DNA of these organisms, and then they put it in a cesium chloride solution, and then it underwent centrifuge, okay, ultra centrifuge. So they spin around the test tubes, and then they were able to separate by density the different molecules found in those cells, okay, found in those genetic materials. So for the first generation, so this is the original parent generation. It's made up of 100% N15 DNA strand, okay? And then after one generation, as you can see, so it's still made up of 100% N15. Then the second generation, you see there that only half of the new molecules have the N15 isotope, and then the other half now has the N14 isotope. So there's now, as you can see here now, that the original parent strand is no longer made up of pure N15 isotope. Then the next generation, there's now more N14 DNA molecule than the original N15 isotope molecule. And then after the fourth generation, there are more N14, and then there are less N15 original parent strand. Okay, so I know it's quite weird to visualize it this way, so I have a separate diagram to help you visualize that. Okay, but this is what they were able to establish. They were able to support that DNA replication was semi-conservative, and that when the double-stranded DNA is replicated, each of the two double-stranded DNA consists of one strand from the original, so this is the red one, the N15, one strand from the original parent molecule, and then the other strand from the newly synthesized or newly created DNA strand. So that's one way to visualize the Mendelssohn-Stahl experiment. So this is another way to visualize. Okay, so they established that DNA replication is semi-conservative. So this is what they started out. The cell exposed to N15 isotope. Okay, so all the parent cells, all the parent DNA are looks like this. But after DNA replication, so this opens up. Okay, this divides into two. So this is where the one parent molecule goes, and this is another parent molecule goes. And then these are the newly created DNA strand, newly copied complementary base made up of N14 DNA strand. So after just one replication, here we can see that DNA replication is already semi-conservative because only one strand of the original parent strand is conserved, the other one is synthesized or created. Now, when the organism undergoes another DNA replication, so this one splits here, and then this one goes here, and then still containing the N14 newly created DNA strand. So another complementary strand is created here, another complementary strand is created here. Same goes for this. This strand goes here, this strand goes here, and then in the middle, there's a newly created DNA strand coming from the N14 isotope. And then finally, this one continuously strips. It goes this way, the other one goes here, and then inside the newly created strand from N14 is the the complementary base strand has been created. So same goes here. So as you can see, after 3 generations, you will have a very small percentage of the original parent strand, of the original N15 isotope parent strand. Majority are now from the newly synthesized, newly created N14 DNA strand. That's why here you will have almost 88% of the generation have the N14 isotope, while only 12% of the generation have the original parent strand. Now, you might be wondering if so what does it mean? So what does it tell us? What does it mean? It means that the parent molecule is capable of creating several copies of itself, preserving itself, even if it takes new materials into the process of making its copies. Okay? They can still accurately create copies of themselves, of the DNA strand, even if the materials are from new strands or newly created strands. Okay? So this is what they were able to find out, that DNA replication is semi-conservative. It preserves half of the genetic material of the original parent strand. Okay? So that's the first characteristic of DNA replication. It is semi-conservative. Then this is the other characteristic of DNA replication. It is semi-discontinuous. So let's break the term. Okay? So continuous, continuous. Discontinuous, there are stoppage, okay, or there are lags, okay, or there are pauses in the process. So there are pauses in the process, but it is semi-discontinuous. So mostly halfway, or mostly not 100% discontinuous. So somewhere in the middle, okay, like that. Because DNA polymerase can only add nucleotides to one end. This results to one strand being synthesized continuously. Okay, so one is being continuously copied. That's called the leading strand. And the other strand being synthesized discontinuously or being copied by fragments. We call that the lagging strand. So how do we visualize that? Okay. So, how does semi-discontinuous DNA replication happens? So, when the double helix of DNA unwinds or unzips, DNA replication on one of the two strands, so that's the 3 to 5 strand, can easily proceed to being copied in a 5 to 3 direction. Now, please note that whenever new DNA is copied, it can only be done in a 5' to 3' direction. Okay, so I will show that later. So this behavior where the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously, this is why we call DNA replication as semi-discontinuous. So how do we visualize that? So this is the original parent strand, okay? The double helix parent strand. Now, in order for it to be copied, similar to what we saw here, okay, what we saw here, an enzyme must unwind or unzip the DNA strand. So that enzyme is the helicase. So the helicase will unzip or unwind the DNA, the parent DNA. So it will have one strand here and another strand here. So you can see half of the green nitrogenous bases are found in this area, in this side. Then the yellow nitrogenous bases are found on this side. Okay. Now, in order for DNA to be copied, okay, DNA polymerase will be the one to use the will be the one to trigger the copying of DNA. So this is the leading strand. It goes from 3' to 5'. In the videos I recommended, it will explain to you what is 3', what is 5'. So it mostly refers to the attachment of the number of carbon in the pentose sugar in which the molecules are attached. So the helicase unzips the DNA, then the DNA polymerase initiates the copying of the strand. So for this leading strand, there's no problem because the copying of the strand happens from 5' to 3' 5' to 3' direction. So this is called the leading strand. So there's no problem here. It will just be copied normally. The complementary bases will be copied naturally. However, for the lagging strand, which goes from 3 prime to 5 prime, they have to do it segment by segment or fragment by fragment. So just one fragment first, and then so an RNA primer will start here, and then this fragment will be the ones that will be copied first. Then eventually it will be stitched together by DNA ligase. Then this part will be copied, and then they will be stitched together. So they cannot do it in one straight strand like this in the lead strand. They have to copy it fragment by fragment and then stick the fragments together. Now those sections of copied DNA, we call it Okazaki fragments. Okay? Now, I've mentioned the base pair several times. We've talked about adenine and then cytosine and guanine, and then the difference between DNA and RNA. Now, just to clarify, these adenine, thymine, guanine, and cytosine are called base pairs. Okay? They are if we visualize the DNA as a ladder, they are the steps of the ladder. Okay? Now, they're called base pairs because they are paired with each other. So, if we unwind the DNA, if we stretch out the DNA, okay, so this, if we look at this part in this way, we'll see that on the sides, you have your sugar phosphate backbone. Sugar, this is the pentose sugar. Pentose because it has five sides, and then this is the phosphate backbone. Okay, it has a phosphorus there, so phosphate group. So they have those on two sides, so they're the same. The only difference is that this side, okay, this sides is read from 5 prime, okay, or 5 prime to 3 prime. Okay, then this other side is anti-parallel. It's on the other side. It goes the opposite direction. It's read from 3 prime to 5 prime. Again, to clarify about those 3 prime and what does it mean, so I recommend that you watch these videos. This is from Amoeba Sisters, DNA Replication Updated. I'll provide the link in the description below. This one also from McGraw-Hill, DNA replication process. They also explain the 3 prime and 5 prime there. So I recommend that you watch these videos. So in the middle of the DNA strands, okay, in the middle, you have your base pairs. So they are paired together. They are always attached to each other. So adenine always pairs with thymine, okay? And guanine is always paired with cytosine. Okay? So to help you remember, the curved letters, the curve of C is also bonded or paired with the curve in letter G. That's it. So they go together. So if there's letter C here, if this is the original parent strand, if there's cytosine here, then a guanine will be paired to it. Okay, so these are the nucleic acids, base pairs. So if there's a thymine here, then an adenine will be paired to it. So just to help you remember, curve sides, curve G, curve C, they are bonded together. Okay, so only with adenine and thymine and then guanine and cytosine pairs can hydrogen bonds be established. So A and T, adenine and thymine, they share 2 hydrogen bonds together. Guanine and cytosine, they share 3 hydrogen bonds together. So these relationships are often called the Watson-Crick rules of Watson-Crick base pairing, named after the scientist who discovered their structural bases. Okay, now this one is very important because when we go to the next video, from DNA to protein synthesis, you will see that the order by which the nucleic acids, the base pairs, are arranged, that from there it will depend on the type of proteins that will be created. So that will be discussed in the next video. So again, watch this video to learn more about the 5 prime to 3 prime concept. So I'll provide the links in the description below. So let's look at this very short DNA strand that will undergo DNA replication and see how it happens. So if these are the base pairs, the nitrogenous bases of the parent or old DNA, we know that the enzyme helicase will unwind them or unzip them. Okay, so they will open up that way. So this is the 5' to 3' parent DNA, 3' to 5' parent or old DNA. So we know that to create or to synthesize a new DNA strand, the complementary base pairs or the complementary pair of the template will be the one that will be the basis. And for 3' to 5', this is how their DNA will be copied. So thymine, an adenine, so thymine pairs with adenine, so an adenine base will be put here. Then adenine will have T here, and then cytosine will have guanine here until you complete the strand. Okay? So thymine, T paired with A with T, C with G, G with C, C with G, C with G, until you complete this new strand of DNA. Now, since the copying will happen from 5' to 3', which is the direction of DNA replication, this is now your leading strand. Okay? So this is your leading strand. It will be copied in that direction. So if this is the leading strand, this is now the lagging strand. So we will start here, okay? Because 3', the complementary base pair is 5'. So this is where the first one will appear, or the first pair will be copied. So G with C, A with T, T with A, T with A with T, and then C with G. So this is the direction from this part, okay? So from 5' to 3' direction. So this is now the other new strand. Now they cannot do it in a continuous manner like this. So they have to do it fragment by fragment, and then they just attach the DNA strands here. So the fragments are called Okazaki fragments, and then the DNA ligase will be the one to attach them together. So this is why it's called the lagging strand, and this is the reason why DNA replication is semi-discontinuous. Now, if you notice, we've talked about this, that in the new daughter strands, through new daughter DNA molecules, half is actually the original DNA molecule or the old DNA molecule, and then the copied ones are the new DNA molecule. Okay? So if you notice, these two daughter molecules or two new DNA strands are actually exact copies of the parent molecule. So if you notice, if you actually go back, okay? Okay, so if you actually go back, this is what we've done. From the original parent strand, we have created these two copies wherein one is the newly synthesized DNA molecule. And then if we continue, this is what will happen to the rest of the DNA copies. We've also established that this is how it happens. The helicase unwinds or unzips the DNA, and then from the 3' to 5', it will be copied directly, continuously. But from the 5' to 3' strand, it will be copied by fragments. Okay? So in this simple experiment, we have established that the two conditions or the two concepts that are involved in DNA replication. One being it semi-conservative, the other one DNA replication being semi-discontinuous. So if you want to practice the base pairings in DNA replication, you could try out this parent DNA strands. So that's the end of our first video for this series. I hope you learned something new today. Don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.